Hi, my name is Jamie, and I will be giving a presentation on mitosis, which is the cell division of an animal cell. The first part of this is prophase, and this happens when the nucleus membrane disappears and homozygous chromosomes pair up. As you can see in the picture, the red and the blue pairs correspond to the different pairs of chromosomes. Crossing over also occurs, which means, as you can see, the chromosomes have a pinch in the middle, and that represents the two chromosomes crossing over each other, making that X shape. And then spindles move to the opposite sides of the cell, as you can see as the orange little squares on either end of the cell. The next phase is metaphase, where the centrals line up along the equator, as you can see, as they go down red, blue, red, blue, red, blue, and the spindle fibers attach to the sister chromosomes. As you can see, there's black lines going to each of the chromosomes in the center, coming from the spindles. Next is anaphase, where the homozygous chromosomes separate and move to the opposite poles. As you can see, half of the chromosomes have broken off from each other, and they're moving towards the spindles on the right and left sides of the chromosome of the cell. They are now called sister chromatids. When they separate, this is called disjunction. Make sure you write that down in your book. Next is telophase, where the sister chromatids reach the opposite sides of the poles, and cytokinesis starts to occur. As you can see, it almost looks like two separate cells are forming now, where half of the chromosomes are on the right side and half of the chromosomes are on the left side. Cytokinesis is the division of the cytoplasm, resulting in two separate cells, and it only occurs after telophase. As you can see in the picture, there's two separate cells now. They're complete with their own set of chromosomes, their own set of spindles, and they have their own cell walls forming, which divides them. The vocabulary that is needed for this chapter is sister chromatid, which is a pair of replicated chromosomes which are identical. They match up completely. They're the same exact chromosome. There's just two of them. Next is spindle fibers, which is the mitotubular apparatus. As we can see, they are here and in here. Next is homozygous chromosomes, which are the same pure chromosomes that are exactly the same that turn into sister chromatids when they replicate. The activity that we will do in class is the class will break up and make a huge circle that will represent the cell. Next, each student, all the students together will walk through the cell division, prophase, telophase, anaphase, all of it, and we'll have people in the middle representing the chromosomes. This will show cells multiplying. Step by step, students will duplicate the cell, and then they will end the cycle by separating into two different cells. This can be done as many times until the groups are small enough that they can't separate anymore. You should have 4 people in your group at this point. 2 on the outsides representing the cell walls, and 2 on the inside representing the chromosomes. Okay, well, I'll see you in class, and we'll go over the process of cell division then. Thanks so much.